Okay, so while people filter in, we're just going to quickly advertise an upcoming BioReam event. So we've got a study group we're hosting next year in partnership with the Quantitative Systems Pharmacology Network. So this is all around pharmacology in respiratory medicine and the interface with mathematical modeling. And we're looking at the moment for clinicians, industry, anyone who kind of falls into this area to submit challenges for this study group, which then a group of, mostly early career scientists will work to solve over the course of the week, or at least, begin to generate solutions for. So if you're not in that category, then please do forward to anyone who you might work with who might be interested. And so with that, I'll introduce today's speaker. So we're really happy to be joined by Ramaswamy Krishnan, who is joining us from Harvard Medical School. So his background is in mechanical engineering, but I'd be hard to sum up everything he's done in his bio, but it is sort of spans academic research and entrepreneurship. Essentially created quite a few devices for sort of in vitro mechanical systems related to the lungs, sort of measuring tissue mechanical properties, and lots of insights, related to asthma in particular. And so I'm not going to do a very good job of summing it up, but hopefully Rama will do a much better job when he presents his talk now. So without too much further ado, I'll hand over to. Oh, and also I should mention, if you want to put questions in the chat during the talk, that's fine, but we'll get to the questions at the end. And also you can answer them, by raising your hand and just unmuting at the end as well. Okay, great enough. So with that, I'll hand over to Rama.
Is everybody seeing my slides?
Yeah.
Excellent. Well, Carl, thanks for the introduction. And there's really no need for a detailed introduction. All I would like to say is I think that this work I'm about to describe sort of encompasses the themes of the BioReam network. I think so. You will hear things on experimental methods, analyses, biology, and so on and so forth. So I think that the audience here is going to appreciate bringing all these aspects together with questions like what I'm about to pose. So thanks again for joining. Let me begin by disclosing my commercial interests. I've co-founded several companies, consult for others, but I want to emphasize that none of this is going to impact the science that I'm about to discuss today. So what I thought, given that mechanobiology and given all of our common sort of interests, I thought I would start with an exercise. And I want everybody in this call, I'm only seeing a few of you on video, but everybody in this call to join me in this exercise. So here's the deal. When I count to 3, I want all of us together to take a single deep breath. Okay? 1, 2, 3. Please. That felt good, right? So it turns out you do that every 6 minutes or so. That's natural physiology. And every time you do that, your airways that can sometimes constrict will immediately dilate. And of all natural strategies your body uses for dilation, that deep breath is considered to be amongst the most effective. However, if you have asthma, somebody with asthma, this effect fails. In fact, in an asthmatic, when they have an attack, a deep breath actually makes it worse for them. Now, this is not my discovery by any means. This has been pursued by several leaders in these fields. And they've identified important roles for airway smooth muscle mass, inflammation, muscle tone, parenchymal tethering, local strains in the airways, stresses in the airways, but a common mechanism has remained elusive. Now let's look at this problem again, but this time let's look at a bronchial airway in a precision cut lung slice, a human precision cut lung slice. So what I'm showing you here is a slice of a human lung. It's viable. The approximately 40 cell types that are in the lung are present in these lung slices, and they're all interacting. This is an airway. this is the epithelium on the inside, there's muscle on the outside. So we're going to take that slice and run an experiment. And I want you to keep your eyes on the airway. First, we added methacholine and then breathing. So what you likely observed is with methacholine, the airway is constricted, but as I was breathing it, it started getting bigger and bigger and bigger. And in fact, if I waited till the end of my talk with this slide, this would return back to its pre-constricted value. Don't take my word for it. I've done this experiment over many different slices, many different lungs, and I'm showing you here average data. On the y-axis is the reversal of maximum bronchoconstriction, and on the x-axis is the amount of stretch that was imposed. Differences between these curves do not matter. Don't worry about it. The message I want to tell you is the more I stretch, the more reversal of constriction you get. Now, all of these data were done with normal, non-asthmatic slices. We're building, and this is now anecdotal evidence from
Sorry,
an
Rawa,
asthmatic.
just sorry to interrupt. Some people are having trouble seeing the slides.
Oh,
can you see it now? I've just because I can see them fine, so I don't understand. I've just unspotlighted Rama in case that was causing a problem. Steve, are you okay now?
No, I mean, I could just switch to your screen now, Carl, with your face on. When Rama speaks, it switched to him, but I can't see. Can other people see this?
There should be tabs at the top that say it's a new Zoom update.
Oh, no, yeah, sorry. I've got it now. It's too modern for me. Got it now. Sorry about that.
No, it's okay. That's a new thing.
excellent.
Okay.
Well, unfortunately, what you missed is looking at your airways inside a lung slice. But I can tell you the crux of it is with breathing, a constricted slice re-expanded. I'm going to now do that same experiment, but this time from an asthmatic lung slice. So first, methacholine, you see constriction, and then breathing. That airway stays constricted. So now you're probably wondering, well, what's going on here? Let's now turn our attention to the airway smooth muscle in those lung slices, and especially contraction of those airway smooth muscles. To measure those forces, we devised a method called tissue traction microscopy. This method requires a substrate that is soft. So by soft, I'm talking about few kilopascal. That's like stiffness of toothpaste. But with toothpaste, when you squeeze it, it just flows away. These substrates, unlike toothpaste, are elastic, which is to say you stress it, the material deforms, but when you release, the material recovers and returns to where we started. So that material is elastic, and this material is also transparent. So in the experiment, you can look through the substrate and you can visualize your airways. What we also do is, and I have to add that these slices are stuck on the substrate. So we synthesize these with sticky surfaces, so you can just slap the slice right onto the substrate. In the experiment, we look through the substrate and we can get pictures of the lumen like this, but we also look at fluorescent beads that we have pre-embedded right underneath the surface of the substrate. So then, when we add a contractant, that lumen narrows. Correspondingly, these beads will displace. We measure that bead displacement with a microscope. We capture those images. We know the stiffness of the substrate. We know the thickness of the substrate. We use a method called traction microscopy, and we can directly calculate those forces around the airways. When we add a relaxant, you can see that the lumen re-expands. Correspondingly, the forces go down. And from every one of these maps, you can get a number to represent contraction. We did this over many lungs, many slices, and I'm showing you here on this left side, the change of contractile force. On the right side, the change of lumen area. First, in response to acetylcholine. So in response, you can see in gray that lumen is narrowing. Correspondingly, these forces are enhancing. When we added a contractant, that narrowed lumen re-expands, and correspondingly, the forces go down. Now, when you look at these side by side, it becomes obvious that these force measurements in black are less variable than the measurements of lumen narrowing. And that's important because we can then, and that's what we're doing now, we can take these force measurements and we can incorporate them in multi-well plates to measure contraction, relaxation changes with many more slices all at once and all at the same time, but fewer slices per condition because the data is less variable. So it turns out not only is the data less variable, but it is also more sensitive to bronchoconstrictor stimuli. So what do I mean by that? In another experiment, we took human slices and we added increasing doses of histamine. In response, you can see that the lumen narrows, forces enhance. But let's look at these small histamine doses. The lumen narrowing is not that, well, it's hardly even measurable, but you can clearly see force changes. So then, all this is to say, we can measure forces reliably. But the bigger question for us is what happens to these forces under stretch? So now let's look at another lung slice. That's the slice, and that's the corresponding contractile force. But this time we will take that slice and superimpose a stretch. This stretch mimics a deep breath. So the protocol is, so it's sort of mimicking an effect of a deep breath. And I want you to keep your eye, I'm going to play a movie, keep your eye on these contractile forces. The first few frames in the movie are prior to the stretch, that's in this region A, and the remaining frames are after the stretch. So keep your eye on these maps. Before, immediately after, and with time. This is quantitation. Contraction on this axis, time. This is in this region A prior to stretch, no change of force, but immediately after stretch, you can see this big force reduction followed by recovery. Now, the obvious question is, what's the mechanism? To get there, we have to go down an additional length scale, and we did measurements now with the cultured airway smooth muscle cell. This is one of those cells placed in culture. Now, one of the things about these cells is when in culture, they're not simply blobs that float about, but they're adherent. And not only are they adherent, but they're also contractile. Shown here are maps, contraction maps. There are arrows here, and these are pointed inwards, and the colors represent magnitude. So this cell is attached at its corners and is contracting the substrate inwards. So let's take that cell and run our favorite protocol. First few frames are prior stretch, remaining are after stretch. And keep your eyes on that contour. Pre, immediately after stretch, and with time. I'm now quantitating these data. That's the contraction over time. In this region A, no change. Immediately after stretch, the forces decreased substantially, and then they recovered to baseline. Now, I've done this experiment, many cells, many conditions, and I'm going to show you next average plots. But I just wanted to say that in order for me to average these data, I took each cell and I normalized it to its own pre-treatment value. And I'm going to show you next those normalized changes. This axis is normalized contraction. This axis is time after stretch. 0% stretch, nothing happens. But even at 2.5% stretch, 5, 10, you can see immediately after stretch a big reduction of force. And in time, in approximately 10, 15 minutes, you can see all these values recover to baseline. Now, while I was doing these experiments, a colleague at the time, and who's now professor at the University of Barcelona, was measuring not changes of contraction, but changes of cytoskeletal stiffness. And he was using similar stretch protocols. The resemblance is pretty impressive. This is changes of contraction, this is change of cytoskeletal stiffness. In fact, if we plotted one against the other, what we observed is that these are strongly linked. These contractile force changes are strongly linked to changes in the cytoskeletal stiffness of these cells. At a molecular scale, we also learned that this relationship is mediated by F-actin dynamics. So first, we fixed cells immediately after stretch. And I'm showing you here F-actin images for cells that are outside our stretched region and within the stretch footprint immediately after stretch. It really was day and night. And if I quantitated this across many cells and dishes and so on, dramatic reduction of F actin. Now, if you fix them here, they're comparable. So the F actin repolymerizes and things recover. We also confirmed this with Western blots. This is the G and F actin ratio. So compared to no stretch, you look at the F actin here, immediately after stretch, the F actin is hardly detectable. But over time, 2.5, 5 minutes, it returns back to pre-stretch values. We went deeper. We started with the actin severing protein gefilin, our favorite protocol, and we compared control cells, shown with the open circles, with gefilin knockdown cells. And what we observed was that compared to control cells, these gefilin knockdown cells relaxed less, but ultimately they recovered to the same pre-treatment value. That is to say that this acute forced reduction after stretch is mediated by gefilin. We next focused on the actin restoring protein zixin. What we observed was that compared to wild type cells, shown in blue, these zixin knockdown cells relaxed to the same extent, but recovered substantially less, which is to say that the force enhancement after stretch is mediated by zixin. Now, I've picked gefilin and zixin. These are two of many, many proteins that can regulate actin cytoskeletal dynamics, which is to say, I've just scratched the tip of the iceberg. We have ongoing work in my lab currently focused on a transmembrane protein called encadherin in the airway smooth muscle that can potentially do similar things and even target it therapeutically to enhance fluidization or to reduce, to enhance this force reduction or to reduce the subsequent force recovery. So from these and many other experiments that we have run, what we have collectively learned is that in response to stretch, the airway smooth muscle fluidizes. And by fluidization, I'm referring to a prompt reduction of force, cytoskeletal softening, enhancement of molecular processes, and depolymerization of the actin network. And it is this fluidization that promotes dilation of that constricted airway in healthy lungs. And we are now testing this hypothesis that in asthma, it is the failure of the smooth muscle to fluidize that is the proximate cause of bronchoconstriction. And not only are we testing this idea, but we're also looking at therapeutics to enhance this fluidization response. So that's part one. And that's everything I want to say about asthma, about contractile forces and asthma. I'm going to switch gears. I'm now going to talk about stiffness in the context of COPD. So first, a brief refresher. Well, why do we even care about lung stiffness? Unfortunately, this audience actually needs no convincing that stiffness can either cause or is a consequence of virtually every lung disease. And that beyond hindering gas exchange airway mechanics, it affects virtually every cellular and extracellular matrix processes, including growth of cell secretion, spreading, differentiation, and even death. And for these reasons, it is a prime therapeutic target. I'll show you some examples in COPD and fibrosis, but it's also being studied in the context of asthma and many other diseases. So then how do we currently measure lung stiffness? These are the two standard methods, the atomic force microscope or the tissue stretcher. In this method, a cantilever beam, you slowly come down and you touch till you probe the cell, you poke the cell, and based on how much you can poke, you can measure the stiffness. And in this method, your tissue is mounted between two loading platens, and you apply a constant force or a constant displacement, and you can thereby compute the stiffness. Problem is that these methods are slow. I mean, you can imagine doing this, it's not fast. They're complicated. How you mount the tissue, how do you load the sample, these are not simple concepts. And they're also low throughput. They're done one dish, one tissue at a time. And neither of these is really suited to be studying lung slices. So to overcome these limitations, we have invented a method called the mechanobiology. So we start with our substrate, and the slices are stuck on our substrate. But then this composite is mounted onto a stretch indenter. And when this gets pushed against the indenter, this membrane together with the sample, they see a uniform biaxial stretch. So we can do this experiment in a single well plate, or we can also run it in a multi-well format. And in the experiment, we observe displacements of these fluorescent beads. Now, if you compare displacements of those beads with sample and without sample, you can get a strain map underneath that sample. From a strain map, and with additional knowledge of thickness of the lung slice, we can solve elasticity equations and we can obtain a stiffness, an average stiffness. So to validate this method, we started first with a material of known stiffness, agarose. We took agarose and we made it at different percentages. And red are stiffness measurements using the mechanobiology. In green and in purple are measurements from other papers with other methods. Both of these authors have used the atomic force microscope or a variant of the device or nanoindenter. And finally, the dotted line is measurement using a rheometer. You see strong concordance. Our mechanobiology measurements are quite, they exactly are, they're really on top of all these local AFM measurements and in agreement with the others. Okay, so now that we have a method that is valid, we turned to human lung slices. And I'm showing you here a representative slice from a healthy and a COPD tissue. You can already appreciate differences in the alveoli between the two conditions. Next, I'm showing you stiffness measurements on a slice-by-slice basis. Each of these circles is a different slice of healthy. This is a different slice from COPD. From these raw data, we can compute distributions and medians. And what we observed was that compared to the normal PCLS, the COPD PCLSs were approximately 50% softer. So you can see, so compare this to the red, the medians. However, this is global stiffness. We next wondered what happens locally at the length scale of the stiffness of a septal wall. To get there, we developed personalized network models. So what we did is we would image these lung slices and we developed code where we can automatically obtain alveolar areas. We obtain these for healthy and COPD tissues, and I'm showing you here their distribution. So in blue is healthy and in red is from the COPD tissues. And then from these distributions, we built network models that represent precisely the same area distributions that we measured. Then what we would do is we varied the stiffness of individual springs such that the stiffness of the network matched the experiment. So, and just to give you a better sense, this is just a magnified view from a healthy, and you can see the larger area spaces in the COPD. For all of this, what we observed was that compared to the healthy, the emphysematous of the COPD slices were softer locally. But there are two important things to note here. Look at these values. This is in the order of 50 kilopascal, 25 kilopascal. So that's an order of magnitude bigger than a global stiffness. So locally, the septal walls are stiffer. However, the stiffness in the COPD tissues is half as much as that of the healthy tissue. So you're probably wondering, well, how do we interpret this? Well, remodeling that happens in COPD softens the local septal walls. So I just told you that in COPD, these walls are approximately half as stiff as in healthy. Remember that these lungs are continuously feeling stretch. Under continuous breathing, these softer walls in COPD will rupture. So that leads to enlarged air spaces. So you have softer walls, ruptured, so fewer walls. Collectively, then this will reduce globally the stiffness of that lung slice. Now, my last slide on this topic, we were also interested in understanding what drives these remodeling processes. So to do this, we did a protein profiling array screen focused on approximately, I think we had about 35 ECM degradation proteins, and we compared them between our normal and our COPD slices. We observed a lot of differences, but I'm focused here on those proteins where the expression was at least 1.5 times greater in COPD tissues compared to normal. So this is the laundry list right here. And I will be honest, prior to the study, actually even now, I don't quite recognize many of these in terms of either the presence or function. But what's exciting for us is each of these hits can potentially be targeted for COPD therapy. Okay, so in the next part of the talk, I want to now focus on IPF. What we did is we took our mechano method, the method of measuring stiffness, and we brought it to IPF tissues. I'm showing you here a representative of a healthy slice and an IPF slice. Green is collagen staining, and you can see these thicker, denser collagen in IPF tissues compared to healthy. Each dot here is a separate PCLS. So this is across 10, approximately 8 PCLS of healthy. This is IPF. And what we observed was, on average, our IPF tissues were approximately 2.5-fold stiffer than our healthy tissues. Now that's global stiffness. Locally, again, as I've mentioned to you, we have technology and methods where we can look at stiffnesses locally. We mapped both for healthy and for IPF. And what we observed was that in IPF tissues, you have these stiffer. So the dark colors here represent stiffer areas. You can see stiffer regions in the IPF tissues as compared to the healthy tissues. So most of these colors are within, this scale, while most of these colors are in that scale. And not only can we map the stiffness, we can also overlay that over autofluorescent maps of those same PCLSs. And from these maps, we can get distributions. And what we learned was that the IPF lung slices had broader distributions, that is shown here in red, compared to a healthy, with approximately 30-fold greater variance than in healthy tissue. So that is to say that not only are these tissues stiffer, but the stiffnesses are also distributed broadly. So you have some regions of less stiffness, some regions of more stiffness, and so on. Now, that's good mechanistically, but the question is, how can we now use these methods, especially in industry, drug discovery, and we've talked about this, and you actually had an introduction slide on this. So we're bringing these methods to drug discovery. To begin, we used a laboratory reagent, just a collagenase to digest collagen in this method to see what would happen. We observed that in response to this collagenase, on a slice-by-slice basis, we observed that globally, the stiffnesses went down. When we looked locally, what we observed was that regions of local stiffness, so originally the IPF baseline, let's look at this hotspot. That was a locally stiff region, approximately 5 kilopascal. But when we added this collagenase, you can see that that becomes light, which is to say it goes from 5 kilopascal down to about 1 kilopascal. So we are able to not only measure global changes, but we can track local regions longitudinally prior and post a drug treatment. But this is just a proof of principle idea with a reagent. We next turned to a real clinical compound, nintedanib. This is one of the 2 approved compounds for IPF therapy. And we said, well, let's put this to the test here. We picked a dose that's clinically relevant, 0.3 micromolar, and we treated our slices for 4 days with this compound. Excuse me. Yeah. I'm showing you here now average stiffnesses in the IPF slice treated with vehicle, so no nintedanib, compared to slices treated with nintedanib. We observed approximately I apologize, I'm not able to see my screens because of these. I apologize. Yeah, we observed that with nintedanib treatment, we observed approximately a 35% softening on average. And here just also is the corresponding raw data that corresponds to these bars. These are the raw data values for vehicle, and these are for nintedanib. But this is global. Locally, when we compared the vehicle IPF slice with the nintedanib IPF slice, we observed that nintedanib treatment showed narrower. So this is the vehicle. Remember, this is the IPF slice with a broad distribution. When we treated it with nintedanib, what we observed was this distribution becomes narrower, and the variance also dramatically drops, an 8-fold lower variance. So why this is important is that not only can this method for, the classic model when, and outcome measurements in drug discovery is often focused on averaged measurements over many tissues pooled together, which is useful for screening, early discovery. But a lot of events happen locally. So ideally, you want to have tools that can not just map global changes, but you can pick local things, local events, and study local interrelationships. And our method is able to do both. So you can get global changes, you can do this in multi-well plates and so on, but more importantly, you can do local. You can look at how your drug, how activity changes things locally. And also by looking at local changes, you're actually making your measurements more sensitive. So for example, in this case, a 35% change of global stiffness, but when you look locally, you can look at how the stiffness distribution is also narrow, how the variance in data decreases. So there is a wealth of information hidden here. Okay, so that's in terms of the mechanics. But I do basic science. We also want to understand what are the molecular origins that underlie these stiffness changes. So to do that, we're bringing our stiffness measurements and we're combining them with omics measurements, such as spatial transcriptomics. This is recent work. I don't have a lot of data to share, but I want to say that preliminarily, we have data where we've done the spatial transcriptomics comparing a healthy PCLS with a fibrotic PCLS. And for representation, what I have done is I have taken a whole bunch of known mechanotransduction genes that change in fibrosis, and I have grouped them together, and their expression, high expressing regions of these grouped genes, is shown in red. So when you compare these two, all these reds, the reds are high expressing groups of these genes. What we observed is, as expected, there is more reds in the IPF compared to healthy. But what's interesting as well is these reds appear to be clustering together rather than being kind of randomly distributed. And this was true not just for mechanotransduction genes, but also for collagen modulating genes. We saw more reds and also more clustered reds. And why that's interesting is this is somewhat reminiscent of those high stiffness regions in lung slices. So our ultimate goal is to take such maps, put them on top of a stiffness map of that same slice, and then we can study not just grouped representations like this, but you take your gene of interest, you can see, does it correlate with a local event of stiffness change? Where does it manifest? Does it cluster? Does it not cluster? How about physiological implications to stiffness? So these are the sort of questions that we are studying, not just in comparing diseases, but also with either drug treatments or with knockdown and so on type of studies. So the conclusion from this part is that we can directly measure stiffnesses. We can measure macroscopic stiffnesses, but also we can go microscopic, and we can do these with disease tissues, and that these stiffness measurements are enabling drug discovery, both in my lab and elsewhere, and it can reveal biological mechanisms. So with that, I'm going to conclude. I want to acknowledge my colleagues, funding sources, and thank you for attending.